Welcome all to today's Tech Talk. Thank you very much for joining. And of course, I'm particularly happy to welcome our speaker today, Professor Gary Bader from the University of Toronto. And he has been a professor at the Donnelly Center since 2006, if I'm not mistaken. And he and his lab are developing computational methods to understand the mechanistics between normal and disease phenotype using molecular interactions, pathways, and omics data. And they also try to work on clinical predictions and identifying therapeutically targetable pathways. So before I hand over to Gary, I just want to make you aware that we stream this event to YouTube, so you can also hop over there if you prefer, or watch it later if you can't listen to the entire talk right now. And aside from that, please keep your microphones and videos turned off during the talk. And for questions, we will have 2 Q&A rounds today because the talk is also split into 2 topics, as you may have seen on the homepage already. So after each, we will have a short discussion round. And for that, please just write your questions into the chat, and then we will ask them after each round. So without much further ado, I want to hand over to you, Gary. I'm looking forward to your talk.
Thank you, Kay. Thanks for the introduction, and thanks for inviting me to talk today. Seems like there's a good sized group of people and audience, and so we can that's suitable for nice scientific discussion. So I have my chat window open, and if I see questions in there that I can answer even during the talk, I'll try to do that. And then hopefully we can have some good conversation during the question session. So I'm going to talk to you today about the general title of my talk is Systems Biology from Single Cell Genomics to Network Data Integration, but I have 2 topics to discuss. Before I get into the topics, I just wanted to talk about the general area of my lab's research. So in general, my group is focused on systems biology thinking. And a lot of work that we've done over time has been trying to understand the genotype to phenotype relationship. And most approaches to study the genotype to phenotype relationship directly try to correlate changes in genotype with changes in phenotype. However, we know that the cellular processes, we know a lot about cellular processes that mediate the relationship. So if there's a mutation, it changes the structure in a pathway, it changes in the structure in a protein, which might change the activity of the protein and its role in a pathway, and that might help explain the change in phenotype, like a gene expression change or a phenotype expression. And so by using prior knowledge, we can potentially gain statistical power in understanding this relationship. And we have a lot of software tools that we develop to help with this as well. It's part of my lab is developing resources, and we work with Trey Ideker and a number of other people to develop Cytoscape, which is the network visualization and analysis software that you probably have heard of. GeneMania is a tool for gene function prediction on the web, and Pathway Commons with Chris Sander and Amek Demir is a central place to store all of this information about pathways that we use in our analysis. And all of the tools are shareable and freely available. So topic 1 is about single cell genomics or single cell transcriptomics, and it's a project that was completed by students in my group, Thin Tran. And the project is about trajectory inference using time series data or transcriptomics data. And the motivation for this comes from thinking about dynamic cellular ecosystems. So when we're thinking about, traditionally when we've been thinking about systems biology in my group, we're thinking about how molecules work inside the cell. But with the advent of single cell genomics, we get a lot of information about thousands of cells per experiment. And so now we can start thinking about how these cells work together. So instead of just thinking about molecules and their interactions, we can also think about cells and their interactions. And there are many tissue-level systems that we can think about now that were not as easy to think about before using high-throughput genomics methods. So just as an example, in development, in the zebrafish, for instance, we go from a single cell stage to a fully formed animal in like 2 days, basically. So it's really an amazing process that generates, can actually work very fast to generate all these interesting structures. So people want to study this. Traditionally, they've studied it with low throughput techniques, and now with single cell genomics, we have more high throughput techniques for studying these dynamic cellular ecosystems. And so in a typical single cell transcriptomics experiment, we get information about many different cells, and these cells have different gene expression patterns that distinguish them into different cell types. And the idea of trajectory inference, the computational challenge, is to order these cells in some usually tree-like structure. It doesn't have to be a tree, that organizes them based on how they might change. So this cell type changes into this cell type and then branches into these 2 cell types. So there are many methods for doing this. Actually, I think there's probably over 100 methods these days. This paper from Salines et al. Has collected these into a nice software package. So generally, the basic idea of all of these tools is to is basically to look at the similarity between cells based on gene expression profiles and organize the cells based on similarity, with the assumption that if 2 cells are close together in this lineage, they're more likely to have similar gene expression, because one cell is the child of the parent cell, and the child should have similar gene expression to its parent. So the way that these tools traditionally work is they take as input a list of genes and cells, a matrix of genes and cells, and the elements of this matrix are gene expression values. This gene is expressed at this level in this cell. Then they apply dimensionality reduction to simplify the problem. There's a lot of correlated genes. And then they build, so each of these dots is basically like a cell type of some kind. And they build a complete graph of pairwise distances between these, and then they find the shortest path in this graph, and that is output as the trajectory, and then you label the cell types, and that's the output. So almost all the tools work with that. So current methods can be improved in many different ways. The 2 ways that we were thinking about improving them is that current methods don't consider time, even though you might have a time course data set, and we have time course data sets with our collaborators that we wanted to make use of. And also, my lab spends a lot of time thinking about how we can incorporate pathway information to help interpret genomics data, and current methods don't use pathway information, so we wanted to think about how we could use that as well. So we developed the Tempora algorithm. This is now published just a couple of months ago now in PLOS Computational Biology. The input, so the way it works is summarized on the slide. The input is a gene by cell expression matrix, as I explained, although this time we have different time points. So T1 is an early time point, T4 is a late time point, and we organize all of the time points, put them all together into one matrix, which is normalized and inputted to the data, into the algorithm. So in our method, we first group the cells by expression similarity. So we use standard single cell transcriptomics methods to identify clusters of cells. Clusters, so if each of these purple circles is a cell, the gray circles around them define clusters of cells that have similar transcriptomes, and usually that represents a cell type. And then the average transcriptome is defined for each cluster. So we do that to reduce the complexity from thousands or tens of thousands of cells to a much smaller number of cell types that we can work with. Then we convert the average transcriptomes at the gene level to average transcriptomes at the pathway level. And we do that by considering all the genes in the pathway. We take their expression level And for a given pathway, we compute an enrichment score or an activity score for that pathway. And so we move from a gene by cell matrix to a pathway by cell type matrix through this two-step procedure. Then we apply redundancy reduction because pathways from pathway databases are frequently redundant. There's lots of versions of signaling pathways, and we know about that redundancy, so we apply PCA to reduce the redundancy of this matrix. And then we do something similar to what the standard trajectory inference methods do, which is correlate the cell types. So we identify which cell types are most similar to each other, and we use the Arachne tool for this. It uses mutual information to compute the similarities, and then it filters the mutual information network to remove for each triangle that's possible. It removes the weakest edge, and that's called the data processing inequality. So that's a standard method that's being used for other purposes like gene regulatory network inference in the past, but we don't use it for gene regulatory network inference. We use it just to compute this sparse mutual information network. Then we look at the cells, each cluster of cells, and we check which cells are expressed at which time points, or which come from which time points. So each of these time points is a different experiment, a single cell transcriptomics experiment, and so we can always track the cells back to the experiment that they came from, and given a cluster, we can check how many cells come from each time point. So let's say we have 100 cells in a cluster, and we can see that 75% of them come from time point 1, and 25% of them come from time point 2. And given that weighting, we basically create a score, which we call a temporal score. So this cluster gets a score of 1.25, and that means that its cells are mostly coming from early time points. Here's a cluster that has cells coming from mostly late time points experiments, and it gets a higher score. So basically the score relates to some inference of time. So the lower the score, the earlier the cluster is found, the higher the score, the later the cluster is found in the time series. Once we do that, we know that cells that appear in later time points need to come from cells that appear in earlier time points, and so we can direct the edges here. So we take this graph of sparse mutual information graph, and we just direct the edges based on going from a low or early time score to a late time score, and then that's the output of trajectory. We visualize it as using a hierarchical layout, network layout, and we place the nodes, which represent cell types, as on an axis that corresponds to this score that we calculated, this temporal score. So we can kind of place them from early to late. And we show this pie chart as well, so you can see how these scores were created. So one of the questions is, how is Tempora implemented? It's a standalone R package. So we wanted to test Tempora. We tried a number of different data sets. I'm not going to go through all of them here, but just go through a couple. There are more in the paper. So one data set we looked at is a mouse brain development data set, specifically the cortex. And this is what the data set looks like. Each of these points is a cell and associated with its transcriptome, and they're coming from different time points. So you can see here that there are, based on different markers, we can identify different cell type populations like progenitor cells and early neurons and neurons. So running the Tempora method provides this output. So we see these progenitors, these APs and RPs are progenitor cells, or like stem-like cells. These are intermediate progenitors, young neurons, and neurons. And you can see the pie charts here colored by the time points. So E11.5 is embryonic stage 11 or day 11.5, all the way to embryonic day 17.5. Because we have pathway information embedded in here, we've used this pathway activity-based way of modeling the gene or representing the gene expression data, we can look to see how the pathways are changing over time. And so we identify pathways that change significantly over time, and the way we do that is we check to see how different the pathway, for a given pathway, how different its activity is over this time course that we infer, the inferred time, as compared to a uniform distribution. So it's very different than uniform. We can calculate a p-value based on that, and then we can just report the most different or most variable pathways. And here are some examples. So how do we know how well Tempora works? So we compared it to some ground truth data as best we could. Thin, manually curated, known developmental trajectories for the mouse brain from the literature, and this is what the literature says happens. These progenitors can make these earlier progenitors can make intermediate progenitors and young neurons, and then so it's actually there's some kind of multi sort of cycles in this graph that are a bit complicated, but so one of the things we learned from literature is that this process is not fully specified. We don't fully know how it works, but this is the best model that developmental biologists have of this process. So we can look at this and compare it with what we got in our Tempora output. So how do we do this quantitatively? So we used two evaluation methods, scores that are in use in the community. So one is the mismatch score. So we can compare two graphs and figure out how many changes we would need to change one graph into another graph. So this is a type of edit distance, and the number of changes is just the score. So if we need to make 4 changes to this graph to make this graph, then that is a score of 4. We also used accuracy measured using the F1 score. So considering also the direction of the edges of these arrows, we can say whether our network matches exactly an edge as well as a direction, and we can compute false positives and true positives and calculate our F1 accuracy. So Tempora pretty accurately predicted this murine cerebral cortex development according to our benchmark. I apologize for the low quality plot here. So the Tempora mismatch score was 1, and the accuracy was close to 100%. So we compared with some other trajectory inference methods. 5 different ones in our paper. So we took 2 methods that were top performers in a recent benchmark paper from the Salines et al. Paper that I mentioned at the beginning of the talk. And so that's PAGA and Slingshot. We chose T-scan as another trajectory inference method to compare to, because T-scan first clusters cells, just like Tempora. You don't have to cluster cells. Many of the trajectory inference methods work directly on cells without clustering them. Monocle 3 is a pioneer method, one of the first and most popular methods, and CSHMM is a time series-based method. So these are the results. This is the actual output of each of these methods, and you can see how the trajectories are organized differently. They're represented quite differently in the different methods. So when we compare the graphs that are defined based on these methods with the gold standard that we defined, we can compute their mismatch score and their accuracy score, and we see that Tempora does much better than all the other methods at both having a low mismatch score, which is good, and a high accuracy score, which is good. So that's on one data set. That was a relatively small data set with only thousands of cells. I forgot to put the number of cells on the slide, but this mouse cerebellar development data set is another data set that has a much higher number of cells, like close to 50 or 60,000 cells, and it has a more complicated differentiation trajectory. So this paper was recently published this time series data set of mouse cerebellum development, the back of the brain, and it also published a gold standard annotated set of what they expect the cell trajectory to be in this system. Tempora does pretty well, but some of these other methods do quite well as well. Some of the methods don't work on such a big data set, and in general, when you consider both accuracy and the mismatch score, Tempora and Paga do quite well here, but they beat the other methods. One advantage of Tempora is that it's got a very fast running time. So Tempora completed this analysis in 60 seconds. It took Monocle 1,700 seconds, and most of the other methods required a very large server to run for many hours, or didn't even complete in the case of the CSHMM, which is not listed on this plot. So we also checked whether the pathway information that we used was valuable. So we checked how well Tempora did with and without this pathway information, and Tempora did better with the pathway information than without the pathway information. So it gets a better mismatch score and a better accuracy with pathway information. So why does this occur? We looked at the correlations that we see between cell types just based on gene expression. And this is a data set that doesn't show it's a muscle data set. And the cells look very similar to each other based on gene expression similarities. But when you look at pathway enrichment similarities, there's a lot more differentiation between the cell types. And so we think that this is causing the improvement of using pathway information. And basically, we think that using the pathway information reduces noise in the data, which is to be expected. Okay, so that was the end of topic 1. I can take some questions now, and feel free to say the questions by unmuting, or I'll read them in the chat window. So the first question is Tempora able to deal with samples from different batches and correct for batch effects in gene expression? So that's a great question. Tempora doesn't do batch correction itself, but what we found, and we discussed in the paper, that batch correction is very important. So if you look at one of these data sets, you can see in this case these cells are all intermingled from different time points. If you didn't so we applied batch correction to get the cells to overlap very nicely. The method we used was Harmony, and that method is being quite successful in our group. We've tried a bunch of different ones. Harmony is currently our favorite. And so in the paper, we recommend that people use the batch correction method to integrate their data. Any other questions?
Yeah, I have another question. If no one else, no, it's okay.
Go ahead.
So with respect to the pathway information, so does Tempora do the pathway annotation, or does that need to happen externally?
Tempora does the pathway annotation. So we basically, my lab maintains a pathway gene set database that we use for pathway enrichment analysis. And we update it monthly. It pulls information from 11 different pathway sources. And Tempora uses that file, that GMT file, and it uses another R package to do the pathway enrichment analysis. It uses GSVA. So it's all packaged in the R package, so it runs automatically.
Okay, and then another question that I had, so in your lab, what type of pre-processing pipeline do you use for generating the gene expression matrix?
For single-cell genomics,
yeah.
For single-cell transcriptomics, we usually use mostly Seurat. The standard features of Seurat are sufficient for most of what we do. We like Harmony for batch correction, and we so if we integrate automatic cell type annotation, we're currently using I think single R, but there are many systems for that, and many of them look good. That's a whole different topic I could talk about, but and if you want more details about any of that, I can let you know.
Okay, thanks.
Any other questions? Okay, so there's a question in the chat. So based on the outcome of the first data, does Tempora work better on one cell type? Neurons seem to be expressed quite heavily, or is it because of the data set? that the development of glia cells are underrepresented. So, so that's a great question and relates to one challenge we have. So with any method that clusters cells, we have to choose the clustering resolution. And in this case, we chose the clustering resolution to match the ground truth that we curated. So each of these cells could be subdivided into different subtypes, and there might be other cells that come up. In terms of glia, I think those are, happen at a later stage of development. Actually, I'm not sure about that. So this stage of development that we're looking for is that we're looking at here in this 11 to 17 day time point is mostly about these cells. So almost all the cells are annotated, and these annotations came from the people, the developmental biology experts that generated this data set. So this is their cell annotations. Yeah, so we can, but I think that was mainly the question about why don't we see some other cell types or not? Why do we see some cell types and not others? And the reason is that's the data that we got, and it was annotated that way. I assume that other cell types are just not present in this data, but there is an issue with cell subtypes that we can discuss more. Any other questions? Okay, so that was a great set of questions. I'm going to move on to the second topic, which is I'll call it unsupervised biological network integration, and we can think about it as having a gene function prediction application. So this is work that is done by PhD student Duncan Forster in my group, who's co-supervised by Charlie Boone, who's a model organism geneticist, and Bo Wang, who is a machine learning expert at the Vector Institute in Toronto. So, so network integration is used frequently for, so it's important because we have biological networks between where we have a lot of relationships between genes. So for instance, protein interactions, genetic interactions, functional interactions, sequence similarity. And these many network types each provide a lot of information that's useful, like gene function prediction based on guilt by association principle. So if you have a gene of interest that you don't know what its function is, but you see that it's interacting in a protein interaction network with many other proteins that have a particular function, your gene of interest might also have that function. And this information comes from many different network types, and these network types don't always overlap well. In fact, most of the time they might have different genes that they represent. The edges are different, and so we generally think of them as complementary, which means that we want to integrate them to get the most information we can. If each network has different types of information about gene function, if we integrate them all, then we can collect all of that information and get a better understanding about gene function through the integrated network. Integration network also can overcome bias and noise in individual networks, which are known. So people talk a lot about false positives in protein interaction networks, and that is one type of noise that's a general problem. So we also assume that some networks are more informative about gene function than others, and we assume that the more networks we collect, the more nodes we collect, the more connections, edges we collect, the better at solving this gene function prediction problem. So network-based gene function prediction has been, so I mentioned the guilt by association principle. That's the main concept that it uses, and there are many resources available. The most popular one is probably String. We make one called Gmania, and there are many others that can integrate networks and create an integrated network that you can use for gene function prediction. All of these methods that exist currently are supervised, which means they globally weight networks trained on some benchmark. Like String, I think it still uses KEGG to weight its networks, and Gmania has an automatic weighting feature that weights the networks based on the genes that you provide. But in any case, they're all supervised, and one of the disadvantages of supervised analysis is that you have to run the supervised, this training step, and that training step doesn't always work well because the benchmark might be incomplete or biased, and you might need to train your data differently depending on whether you're talking about the one protein function or another. So ideally in machine learning, and this is sort of a typical problem for any kind of supervised machine learning. So in the field of machine learning, there's always a drive towards figuring out unsupervised methods. So unsupervised methods like clustering don't need a training data set. They just identify structure in the data, and they do that automatically, and so they're basically automatically learning something about the data. So there have been some unsupervised methods, I mentioned them here, for network-based network integration. Typically these work by trying to preserve lower dimensional patterns like clusters or modules, which in a protein interaction network could be protein complexes, and in a functional interaction network might be pathways or modules or systems in the cell. And so most of these methods, these unsupervised methods, identify these clusters and then try to match them up. There's different approaches to this. Matrix factorization is a popular one, autoencoders, and also SMF uses edge reinforcement. So if it sees the same edge multiple times across different networks, it keeps it and then eventually integrates the network that way. So we developed a method called biological network integration using convolutions, or BIONIC. The method is a deep multimodal autoencoder using graph convolutional networks. I'll explain what that means. It has some nice features. The most important one is that it's scalable compared to other methods. It also does well on benchmarks, as I'll show you. So how do we train a network without a benchmark? And in this case, how do we train the deep learning network, the neural network, without a benchmark? So we use technology or an approach called an autoencoder. So in our case, we have input networks, and we encode them into some low-dimensional space that captures it's intended to capture the essence of the input, like, for instance, the modules that are present in the networks. And then we can decode those into an output and create output networks. You can represent these networks as matrices, and then we optimize all the parameters in this neural network. So these boxes represent many layers of neural networks, and we optimize the system, select parameters such that the neural network can do this processing and get output networks that are as close a match to the input networks as we can get. So we optimize so that the output matches the input, and that's how we can train the network without a benchmark. So it uses its own input as a benchmark. You can think of it as that. But because it goes through this low-dimensional hidden layer, it's trying to capture some essence of the input that is good at capturing the essence of the output and matching it to the input. Okay, so now how do we get the networks, the protein interaction networks, into the neural network? So traditionally, people have encoded networks using, so they've converted networks to vectors, and vectors are needed as input for the deep learning method, using some kind of pre-processing step. Traditionally, node to vec is a method that does this. What we wanted to do with BIONIC was, so the disadvantage of this method is that we rely on feature engineering. So node to vec decides how to convert networks to vectors, but we might want to learn different ways to do that depending on the network. And so there's a new technology in deep learning called graph convolutional networks. So it's able to take graph or network inputs directly, and that graph convolutional network is part of the whole neural network learning system. And so it can convert the graph to vectors internally, and that is basically incorporated into the learning process. So instead of doing it the same time each time with a feature engineering approach, we let the learning system figure out how to best convert the graph to a network, the graph to a vector. So here's sort of a representation of this, where each node is represented as a different value in a vector, and this is what we kind of combine it with an identity matrix, and then the nodes kind of share their information back and forth with some kind of can think of it as a propagation step. And then there's dimensionality reduction. They get forced to encode the data into a smaller number of dimensions. And then this matrix here represents the learned features, so this low dimensional space. And then we take this and we decode it back to this space to compare it. So the low dimensional space, this is sort of a bit of a zoom in on an overview. Okay, so this is a zoom in on how the graph convolutional networks work. And this is an overview of the whole method. So we take these networks as input, we convert them to their adjacency matrix forms, we put them into the graph convolutional network box. The output is a low dimensional embedding of each graph, each of these networks, and then we combine them inside the system to get integrated features. So this is where the network integration comes from, but we integrate at the low dimensional feature representation of each network. And then we output that, we convert that back to a matrix which we compare with the input, and we minimize the difference in an iterative fashion. That's the autoencoder process. So this is sort of an example of what, if we put in a lot of yeast protein interaction and genetic interaction and other types of interaction data, we get a low dimensional representation of all the genes and their relationships. So here, this is what it would look like in sort of a 3D model. In this case, it's a 256 dimensional space compressed down into, for visualization, into a 3D space. I think we actually use 512 dimensions in our current version of BIONIC. Okay, so how do we evaluate that this is working? So we tested on yeast, Saccharomyces cerevisiae. We have 3 networks that we chose as a starting point. So one very large genetic interaction network, and one large protein interaction network, and also a co-expression network that was also quite large. It looked at hundreds of gene deletions and had hundreds of co-expression experiments. Okay, so we compared BIONIC to 3 integration approaches: a naive union of all the networks, just combine all the nodes and edges across all the networks. We expect that this wouldn't do so well because there might be a lot of noise, especially like in the co-expression network, and so we just combine all the noise together. So, but we thought that would be a good baseline. ICEL is a matrix factorization approach, and mashup is a diffusion state approximation method. But it's basically both of these methods work. Like I mentioned, they've tried to integrate networks by identifying low-dimensional features like clusters and matching them up between networks, input networks. Okay, we have 3 performance evaluation criteria that we used. So one is trying to find out if when we learn the features, are the genes that are close together in the learned feature space more likely to be co-annotated in Go biological process, known complexes from Intact, and pathways from KEGG. We use precision recall as a measure, and then we plotted all of the different methods here. So the average precision is measured over all of the different terms that are in these benchmarks. So the take-home message here and in general for the talk is that BIONIC does very well. One interesting thing that we found is that the union does well and actually beats out some fancy methods, like ICEL doesn't usually do as well as the union, although mashup can do quite well. We tried a second performance evaluation method is module detection. So taking the output, this low dimensional embedding, and clustering it using hierarchical clustering with a lot of hyperparameter tuning to try to globally optimize clusters that come out against the benchmark. So say we have a benchmark of intact protein complexes, we cluster our output data, and we match the clusters to the intact complexes, and then we can try lots of different parameters of clustering, and we choose the one that where globally all the clusters that come out have the best overall match to the benchmark. So that's what globally optimized on the benchmark means. And then we compare with our known modules with the adjusted mutual information measure, and over all modules, we can see how well these different integration strategies are doing. And again, BIONIC does well and beats the other methods. Yeah, so that's the same roughly here. Actually, in this case, the union does very well for gene function prediction. So in this case, we take the features that we learned, which are, say, this 512 dimension space. So each gene has a vector of 512 values associated with it. That's the learned representation of that gene in the space of all functions. And we use a supervised machine learning method to say, take those features, those vectors as features, so for instance, logistic regression, and try to classify it based on known functions. So train it with some function, test it on a held out set of functions, and then see how well it does with an accuracy measure. And that's this last benchmark that we ran. Okay, so again, the take-home message is that BIONIC does well. It's not surprising. We tried looking at how well it does compared to these other methods on a range of different function categories that are high-level function categories from the genetic interaction network paper that we use. And the take-home message is that in general it does well, but in some categories it does well, very well, in some categories it doesn't do well. And none of the methods do well, for instance, on some of these categories. So that's good to know, because it's not a magical solution that does well for everything. Here's an example of the integration. So here's the proteasome from the 3 different networks that we used as input, and the BIONIC result, just to check visually of how well it's working. The most important thing, I think, right now for us is that BIONIC scales well compared to other methods. So if we try to give these methods an increasing number of networks, like yeast co-expression networks, what we find is that all the other methods, other than just the union, don't scale very well. So, and in particular, mashup, which is a pretty well-performing method, cannot integrate more than 7 networks or so, large networks. The authors of the mashup paper, interestingly, knew this and provided an alternate approximate solution using singular value decomposition. But when you apply that performance of that method is terrible. So you should never use it. It was not something discussed by the authors, but in our evaluation, it's just clear that this approximation method is not as good as the actual method that is published by the authors. So once you get to a higher number of networks, then BIONIC really starts doing well. So its performance improves. It's much better than union, and the other methods don't even work. Like mashup can't compute, and or the other methods have poor performance, and they continue to have poor performance. We can also look at the scaling based on the number of nodes, and we get the same basic answer that the more data we have, the better we're doing in our performance on our benchmarks. I don't show all the benchmarks here. We're still working. We're hoping to submit this paper soon, and so it'll be in bioRxiv. But for the purpose of this talk, I'm just showing a small number of benchmarks here. And in this case, we had input networks from 4 human protein interaction networks. Okay, so one of the assumptions that I talked about at the beginning of this topic is that more networks are better. And in general, especially from a supervised machine learning point of view, like string resource or GMania, I think in general it's safe to say that the field generally assumes that the more networks you put into these tools, the better it will do. And we wanted to check that assumption. So when we looked at using 3 networks with BIONIC or 68 networks with BIONIC, what we could find is that we could find a set of 3 networks that actually did better than the set of all 68 networks that we collected. So if you look at these values here, like in module detection, the score is better. It's above 0.2 here, and it's below 0.2 here. when we have fewer networks. It's better when we have fewer networks. So that's contrary to what we expect. Now, we chose these networks to be the best 3 that we could find. And if we just randomly choose 3, we get similar or worse results, mostly worse. So, but it's interesting to note that more networks is not always better, and this is something that we need to think about. So some networks are more informative. So if we look at all these 68 networks in different categories, genetic interactions, protein interactions, co-expression data, we can see that genetic interaction networks do really well on Go biological process benchmarks. Protein interactions do well on complex benchmarks, and co-expression networks do well on the KEGG pathway benchmark. So that's also good to know. So another thing we noticed is that not all of the complexes that we predict if we take the output of BIONIC and cluster it to find protein complexes, many times, and I'll just take you through one of these plots here. So let's say we looked at BIONIC versus the genetic interaction network. And each of these circles represents the score, the match score or performance of finding a known complex. So each of these dots is a known complex, and if the score is away from the diagonal on the BIONIC side, it means BIONIC did better at predicting this complex given the benchmark compared to the genetic interaction network if we used it by itself. And so you can see that, for the most part, BIONIC does well. This is sort of a distribution of all the module scores. So you can see that BIONIC is doing, with a score of 0.48, an average performance of 0.48 is doing very well compared to all these other methods. But not every module is working well. So sometimes even in this case, there are complexes that the genetic interaction network scores as perfectly, finds perfectly, and BIONIC doesn't do very well on. So one of the future directions for us is to figure out if we can improve so that the main future direction for Duncan, who's doing this work, is to continue to work on the BIONIC integration method to figure out how to get it so that either we can just give it all the networks, we don't have to somehow figure out how to pre-select networks to get better performance, and also when we do that, we always use all the information from all the networks to improve the performance so that all of the modules, for instance, would be better or equal than any of the input networks. So there's still major challenges in this field, even though we're doing better than, we're making some advance. There's interesting research challenges ahead. So with that, I'd like to thank, I thank Finn and Duncan, who did the main work on the projects that I mentioned, and these are members of my lab, past and present, who work in different areas, and we get funding from mostly Canadian funding sources. Gordon Keller, Stephanie Pratza, and Frida Miller were people that we interacted with a lot on the Tempora project to provide us with single-cell, time series-based single-cell transcriptomics data. So thank you very much for your attention, for all those details, and I'm happy to take questions on anything in the talk.
Yes, thank you so much for the interesting talk. Yeah, please post more questions you have, or you can also speak up if you prefer. I actually have one question. So I think on slide 34 or so, you had this matrix with some networks that could not be predicted by any of the methods. Do you know why that is? Like why some networks are trickier than others?
Here?
Yeah.
So these are functional categories that are trickier than others. So, ribosome biogenesis, we do very well on, and mashup does very well on. The main reason for that is because the ribosome biogenesis function incorporates things like the ribosome, which is a very strong complex that's identified clearly as a module or cluster in all the input networks. And one of the main features that the autoencoder is learning ends up being modules. So if our input data doesn't have good modular structure in a particular class of function class like DNA replication repair, if we visualize that network, it would likely be more sparse. And it's actually a good idea for me to show that visualization in these talks, but to answer your question. But that's the main reason why we think it's not working. It's not just modules that we think that these systems are learning. They can learn other aspects of the other features of the input graphs, but that's the one that's easiest to think about, I think, and I use it as an example. It's also one of the ones that's the most strongest signal in the data.
I see. Thank you. Yeah. Sven?
Yeah. So thanks for the talk. Really, I think it's really nice tools. Basically, my question is also partly triggered now by the previous question by Rikke. What you just reported on Friederike's question is literally an explanation for the prediction or for the missing prediction. Then my question would be like, explainable machine learning, is that something that you have in mind also? Do you provide explanations for your predictions or not routinely?
So that's a great question, and it's a super important topic in machine learning. In the BIONIC project, we're not addressing that currently, but we have other projects in the lab, and actually my colleague Trey Eideker, who we work on the Cytoscape project together, his lab focuses a lot on that. And the other way of thinking about it is interpretable machine learning or visual machine learning. So there are many different approaches to that. We're not using any of them now in BIONIC, but for example, you could use, instead of just providing all the data as vectors to the machine learning method, you could use you can engineer your features so that they use interpretable features. Like instead of just throwing a lot of genes, you can provide pathway information as the input or pathway activity scores, like in Tempora, which then afterwards it's kind of easier to sort of see which pathways are providing a lot of information that's different than like a uniform distribution. Trey's done this interesting work called D-cell, where he creates a neural network that takes the structure of the gene ontology exactly, and then uses it to predict phenotype from genotype, and found that it works. And then you can see what the weights are in all of the different neurons in that system, which correspond directly to gene ontology terms, and you can get a sense of what's important or how it's making the prediction. But that's a big research problem that's mostly unsolved. It's a very interesting discussion.
And yeah, thanks a lot. Of course, that's also an interesting research issue that we are also facing here, the explainability. And another question would be how now, obviously, for the tool development and performance evaluation, you used a lot of publicly available data from a multitude of papers. Do you also have ongoing applications with domain colleagues where your method did provide new insights as compared to existing?
Yeah, the motivation for all of these methods comes from working with biology collaborators. And in BIONIC case, Charlie Boone, who's a yeast geneticist, is generating a lot of this data. Actually, the genetic interactions come from his lab. And his lab's goal is to generate a map of the cell that we can learn more about the function of all genes. So that's why we're working on. So that's the reason why we're working on this project, is to generate the best map of the function of all genes. And then he tests hypotheses in yeast experimentally in this group, and we have other projects that stem from that would be separate talks, predicting other types of functions and interactions that they're interested in. Yeah, so most of the work in my lab is motivated by collaborations with people that are generating data that we're working with.
Yeah, thanks a lot for the interesting talk. I really like the graph convolutional network approach. And I was wondering if you are also testing this approach for other kinds of network data, maybe gene expression pathways for other projects, or?
Yeah, so the next phase, so we just got a new student who's going to start soon, hopefully, and his job is going to be to try to incorporate as much types of networks as we can. So the first thing he's looking at is protein sequences, and it's actually doesn't have to be networks. We can integrate into BIONIC different. We don't have to use graph convolutional networks. We can use that for all the network data, but ideally we would use the whatever data we have. So protein sequences as kind of strings. The gene expression data we're converting to networks, but it would be nice if we could just input the matrix form of that. But that would not use a graph convolutional network. It would use a regular convolutional network or some other type of input, and we can put those all together. So instead of using graph convolutional networks here, so we use one graph convolutional network for every input network, and so you can imagine there can have many of them here. We could put other things here, like a convolutional network for images or something like that. I don't know if that answered your question in a kind of more broad way, but yes, we're thinking about lots of different types of input to see what this can handle.
Yes, thanks a lot.
Did that answer your question?
Yes, thank you very much.
Yeah,
Yep.
okay. Okay, I think we're nearing the hour, but are there any other questions?
Well, it doesn't look like it currently, but I'm sure we can also write you if more comes up.
Yes, happy to answer questions by email and discuss in the future anything of interest to people. Yeah, thank you very much for the great questions and attention.
Yeah, thank you so much for the talk. I will close the stream now, and if people want to stick around for a couple of minutes to